What, you thought I was going to stop with the music? You thought I was finished? I'm never finished. I am never finished. Welcome back, y'all. As promised, I'm a man of my word, giving you another video. This time we are finishing up our protein synthesis journey, and we're going over the second step, which is translation. So let's recap really quick. I know you just watched the video and you couldn't wait and you came to this video, but just in case you forgot, transcription, that's where we have our DNA double strand, and now we have created a single strand of mRNA. Now we're taking that mRNA and we're going to actually translate it into a protein or a polypeptide chain of amino acids. Let's us get started. So, following along with our handout here, you should be on the backside where it says translation. So at the very top, what is translation? Well, I just told you. We take an mRNA and we turn it into a protein, baby. That's what we do. That's what we do. Now, this is not happening in the nucleus. You know why? Because DNA can't leave the nucleus, which is why we have to create the mRNA. This is actually taking place in the cytoplasm. Be more specific, it's happening inside of ribosomes that are free floating around in the cytoplasm. So the first step of translation, we have the ribosome or the rRNA that reads the mRNA strand, right? That's the first step. The second step is transfer RNA comes on over with the amino acid and the matching codon to match mRNA. That is step number 2 of translation. Step number 3, I just revealed to you a new vocabulary word, codon. See if I can get my pen to work in this video. It didn't work in my last one. Hopefully I can get it to work in this one. So let us go here. Pen work. Nope, still not working. So we have a codon, right? This is our codon. That is on mRNA. tRNA has what's called a anticodon. All that means is it matches to whatever the codon was to mRNA. Again, perfect example down here at the bottom. You have your mRNA with the codon. tRNA has the anticodon, which matches that base. So for this base, it would be UAC because this goes here, A with U, and then C with G. That's what happens with step number 3. Step number 3 also brings in something very important that we need to know about. It is what's called a codon chart. This lovely chart, take a look at it, stare at it as much as you need to. Because guess what? I'm making a third video specifically going over codon charts. I may even bring my Steve Harvey mustache back, and I may be Steve Harvey for this video. You never know what I'm going to do. But the most important thing I want you to take away from this codon chart is understanding how to read it, because I know it can look super confusing. We're going to start all the way here on our left, and you're going to read that first letter first. So if our first letter, if our codon is A, or if our codon is U, C, G, we're going to start at this first letter. So we know we're going to be in this section, right? Second letter along the top, C. So we're going to be here. We're going to come down, and then we're going to come all the way over to the right column, and we're going to find G and come across. UCG puts us at the amino acid serine. That is how you use a codon chart. What do you think happens now? Somebody say it. You know it. I got to make sure you got some time for some practice. So on this next slide, I want you to practice with this set of mRNA. What would be the matching amino acids to go with it? I'll give you 1 minute. Let me put my music back on. Matter of fact, I'm going to give you 30 seconds because I know you're going to get the answer really, really quick. While you do that, just think about it. Think about it. Don't think too hard though. All right, I know you got the right answer, right? Of course you did. Here is what it looks like. These are the amino acids it codes for. As you can see, with U, our first letter here, I'm sorry, we started at A. With A, our first letter here, then we have U as the second letter, and then G is our start point. So you will see a lot of times you may see a amino acid that says start and you have a stop signal. We'll talk about that in later videos, especially when we get into codon charts. But either way could be right. If you wrote out MT or MET would be fine, or if you put start, that would be fine as well. But the one thing I want to make sure is that you understand how to read it. Now, codon charts don't always have to look like a square. You may see a codon chart that looks like a circle. And man, I wish my pen would work because then I could draw it for you. But I'm going to try to do it with my mouse. Let's see what happens here. You may see a circle that looks like this, and another circle, and then a little circle in here. You read it the same way you would the square. First letter here, second, third, and then on the outside would be the amino acid. Again, I'm going to make sure you get plenty of time to practice reading the codon chart, not only in videos, but make sure we do it in class as well. And that leads us almost to the end of our journey in protein synthesis. It takes us to the fourth step. Now we get those amino acids linked together. Now we have a protein, or what we call a peptide chain. Peptide, I know everybody remembers from unit, was that 1 or 2 when we talked about biomolecules. This is just a long chain of proteins. Actually, it's polypeptide if we want to be extremely correct. Peptide is the singular version, right? So polypeptides, that's what we create, and that is our proteins. That codes for our genes. This part, just telling you a little bit more. We've again went over ribosomes, what they do. They read the mRNA. They can also be attached to the rough endoplasmic reticulum. Again, something we learned in units 2 and 3. Or they could be free flowing, and then protein leaves through the Golgi apparatus. Oh. Gene regulation. Don't get too caught up on this. The most important things to know on this is that there are regulators in your body, what we call gene regulators, that regulate certain traits or certain proteins being turned on or turned off. Gene regulation only happens during transcription, which is the what step in protein synthesis? The first step. You got it right. Oh, thank you so much. I was getting nervous there. I didn't think anybody was going to answer. Yes, this only happens in the first step of protein synthesis. Cell differentiation, right? This is when we talk about cells being different or specialized. During transcription, because we just went over, that's when gene regulation happens, and cell differentiation is just another part of gene regulation. Your cells start out as stem cells. Depending on the code that they get, determines what type of cell they're going to turn into. So these stem cells can be turned into, or can have the ability to turn into many different type of cells. What you need to know is, again, this is going to happen in transcription. And when we talk about cell differentiation, we're just talking about cells being different, or what I like to say, cells being specialized, right? And our last and final point is just a review point on the central dogma. We went over this on Monday, but if you were not here on Monday and you missed it, the central dogma of biology, or molecular biology, just states that DNA is transcribed into RNA, and then that RNA is translated into a protein, and those proteins are what make each and every one of us different, right? In transcription, we're getting the instructions on how to make a certain protein, and now in translations, we actually in the kitchen and we whipping it up, and now we're using that instruction that we've got to actually make the protein. Remember, transcription happens in the nucleus because DNA can't leave the nucleus. So there has to be a copy that's made inside the nucleus, which is that RNA, which is now being translated in the cytoplasm at the ribosomes, because that's where it goes down at, right? This has been another video for you. I hope you found this video helpful. If you need to, go back, watch it again, tell your other friends in other biology classes, tell them to subscribe to the channel. I'm putting more and more content out for you guys to help you out as much as I can. And remember, Mr. Scott enjoys his classes equally. I don't have a favorite class. Not, I love them all. 7th, 4th, 5th, 6th, 7th, 8th period. I enjoy them equally. You guys have a great day. Subscribe to the channel. Like. See you next time. Peace.